Okay, our next speaker is Professor Stefan Green. He's an associate professor in Rush Medical College, Chicago. He's also director of the genomics and microbe core facility there. He's interested in the interaction of complex microbial communities. Today he's going to talk about effects of space flight on the mammalian gut microbiome.
Good morning. There we go. Good morning, everyone. Really happy to be here. My big thanks to the organizers. I want to do something a little bit different than and talk a bit about some specific mission on the twins astronaut study that NASA conducted a few years ago. I will warn you that Fatih said on one of his slides, you should always have an N greater than 3. So this is the worst case scenario for doing a study. There just aren't that many monozygotic twins who are also astronauts. So we have an N of 1. So as I present my data, I just urge you to remember this is an N of 1 study, and there's a limit to how far we can extrapolate the results of such a study. Now, we do have mouse mission data as well. I'm only going to touch on it very, very briefly, just due to time constraints. Okay, so I think all of us are interested in general on the effect of spaceflight on our overall human physiology. As I'm a microbial ecologist, we're particularly interested on the host-associated microbiome, and in this particular study, the gastrointestinal tract, as measured by analysis of fecal microorganisms. So some of the preliminary data have shown that there is an effect of flight, but we're still not quite sure how robust those effects are. And there's a big knowledge gap between knowing that there's a shift in the microbial community and what that means just for total gene content in the gut microbiome, what it means for the activity of the organisms, what genes they're expressing, what metabolites they're producing, and ultimately the effect on host physiology. So we have a lot of gaps. And so NASA is of course, ongoing missions to study the gut microbiome and other microbiomes in mammalian models. And so I just want to, again, two of the major missions that I've been involved are one with the NASA twin study and also some of the rodent research missions. Only going to really talk about the twin study here. Okay, so for those of you who are not familiar with the twin study, there are two astronauts, Mark and Scott Kelly, who are monozygotic twins, both of them who are astronauts. And the mnemonic to remember which one is which is Mark has the mustache, so M and M, and he's the guy who stayed on Earth. And Scott, who doesn't have the mustache, went into space, so S for went into space. So of course, they've both been in space, but Scott Kelly was basically in the space station for about a year. And at the same time, they thought, hey, this is a great opportunity to do multi-omics analyses of twins, both of them who, again, share obviously genetics, but also shared experience in space flight. So, okay, I know I only have 15 minutes, but that seems to be a bit ridiculous. Let's go back here. Sorry for that. So I will just say that I'm the microbiologist associated with a study that's really run by Fred Turek and Martha Vita-Turna at Northwestern University in Chicago. They're the lead investigators, particularly interested in circadian biology and the effects of spaceflight on disrupting the circadian rhythms in mammals, and again, what does that do for our gut microbiome? We have multiple collaborators at my current institution, Ali Keshavarzian, who's a gastroenterologist, and then I used to be at a different institution 2 blocks away, and so we have multiple members of the laboratory there doing wet lab work and also helping with the bioinformatics analyses. So I don't want to belabor this, but if you ask a question to almost any scientist and you ask them, is spaceflight good for humans? And I think the unequivocal answer is that it's probably bad. There are all sorts of unpleasant outcomes. We already heard about the headward shift in fluid. That also affects ocular issues, and you get vision issues that come from living in microgravity for a long time. Of course, there's very well documented bone loss, bone density loss, and muscle mass loss. But I also want to point your attention to another bad feature of space, which is difficulty in sleeping. Okay, it's not just that you no longer have light dark cycle that you expect. But also, I just want to show you what a bed looks like in space. So this is when an astronaut, it's like a little, I don't know, hamster hole that you got to like Velcro yourself into and all your stuff. It's not really well talked about, but it can be very difficult to get a full night's sleep in microgravity. I will also point another feature that's not very pleasant in outer space. This is a toilet, so you can imagine enjoying using this while also basically weightless. So there are logistical challenges also to doing things that are totally easy for us here in a strong gravitational field. Okay, so as I mentioned, this is an N of 1 study. We had a very, very simple sampling strategy, and part of this is really to accommodate, the astronauts here. This was not again, N of 1, not a rigorous experimental design in one sense, but we wanted to get both baseline samples, and again, remember, this isn't just microbiome. There are 10 teams involved in this, assessing a whole wide variety of omics and also psychological testing and cognition testing, RNA-seq, epigenetic analyses. I'm just focusing on one little corner of the full study. But for the microbiome aspect, we have basically a simple strategy. We have 2 baseline data points prior to launch. Then during launch, we have four data points. And then post-launch, post-return to Earth, we have three data points. And from Mark, again, mustache on the ground, we have the same number of samples at roughly approximately the same time. Now, when I got involved in the study, the first thing I was asked, I'm like, obviously they're going to be eating the same thing, right? And they're like, you crazy? There's no way Mark Kelly is going to be eating the same thing as, dehydrated food at the same time as brother. So that control we don't have. So what we really need those baseline samples as a way to normalize the microbiome of each individual in the study to their own baseline and to look for kind of effect sizes and changes from baseline. And again, just a reminder that Scott was in space for almost a year, and really the longest single stay for an astronaut. Okay, as I mentioned, we have multi-omics analyses, and the microbiome was just one piece of this. From the microbiome perspective, I didn't want to do too much. N of 1, not well controlled in terms of diet. There's a limit to how many questions we can ask from such a data set. So we had basically very simple questions. Do the twins have the same microbiome at baseline? They're twins, maybe they eat similar things, maybe they have very similar microbiomes. I wasn't too hopeful, but we got to ask that question. More interestingly is, do we see a loss in microbial diversity while the space twin is in space? Okay, this is really the kind of heart of it. If you lose microbial diversity while in space, it's extraordinarily difficult to reconstitute it. Okay, if you're going to Mars, you're gone for 3 years, you can't walk in the forest and take a deep breath of fresh air and get inoculated with fresh microorganisms. And so it's a challenge if we start to see loss of microbial diversity. In a similar way, for example, if somebody gets an infection and needs to take antibiotics, we could experience a loss of diversity. Could be very difficult to reestablish. So a key area for us. We also want to know, do we see a consistent trend in a shift in microbial community structure of the microorganisms in the gut during flight, in the flight twin, that we don't see in the ground twin during the same time period? Okay, again, N of 1 study, you have to be a little bit creative on how you're going to analyze the data. And then also we want to see, do we see upon return, if there are consistent effects in shifting the microbial community during flight, do we see a return basically to baseline after return to the ground? And then, of course, we're interested in pathogens. There's evidence from other studies that the pathogens can increase growth during microgravity flight conditions. So we want to know, does that also pertain in a host-associated microbial community like the gut microbiome? Now, in the interest of time, and so there's no mystery here, I'm just going to give you the answers. And we can just say no, yes, somewhat, and some. Okay, so I still have time, right? Okay, all right, great. So we can keep going. All right, we'll dig in a little more. Just a very tiny bit on methodology. We do, the astronauts have swabbed stool samples. So we're not getting actual stool, it's a swab and freezing of the sample. The samples are frozen and then returned to Earth when possible. We do DNA extraction. And then in this particular study, other studies, we've done 16S ribosomal RNA gene amplification. We know that there are bias issues, as Nikos addressed in his talk. In this study, we use shotgun metagenome sequencing, which has somewhat less bias. And I'll just also mention one feature that the kit that we use is also capable of targeting single-stranded DNA, allowing us to get a broader range of DNA viruses as well if they're. Okay, so let's ask our first question. Do we see similar communities in our two twins? Do we see similar levels of diversity? So this is just kind of a genus level profile of the ground twin across all the nine data points that we have. And then we have the same 9 data points for our space twin. And if we just run some basic statistics, we can say, hey, these have different diversity consistently across all the time points. And also, if we look at beta diversity rather than alpha diversity, the communities are consistently significantly different from each other. So not really a surprise, but the twins are different, okay? They might be monozygotic twins, but they don't have the same microbial community at baseline. Of a little more interest to us is within Scott's microbiome, do we see any effect on global diversity? So we're talking about alpha diversity here within the 4 flight time points relative to the 5 ground points. Now, again, this is a kind of a limitation. We do a 4 versus 5 comparison in which the 5 contains both the baseline and post-flight samples. And basically, we have a nice answer that no, in this particular case, we see essentially no effect on overall alpha diversity in Scott Kelly's fecal microbiome, which is very positive. Again, N of 1, but it shows that at least it's possible to be in space for a year and not lose microbial diversity. Okay, but do we see any shifts in the structure or the composition of the microbial community? And here we start to see some interesting effects. We're looking both at the Shannon index, which is an alpha diversity index measuring both richness and evenness of the microbial community. Those are down here. We basically don't see much change in either the ground or flight twin. But within a comparison of 2 dominant phyla, so the formerly known as Firmicutes and Bacteroidetes phyla, which are in general mammalian fecal microbiome, are the most abundant, we see a dramatic increase in the ratio between the Firmicutes and Bacteroidetes during the 4 flight time points, and that returns to baseline after flight. So very interesting. We see a discrete shift in microbial community. And what's interesting is in our mouse studies, and again, this is the only slide you're going to see on mice, and just by the way, down here are these fancy mouse cages for sending more than 1 mouse into space. We see our Firmicutes to Bacteroidetes ratio, and this is in red our flight animals and in ground animals, and we see the exact same trend, increase in Firmicutes to Bacteroidetes ratio. So broadly, when we look at the beta diversity, so again, a comparison of the microbial communities from every sample to every other sample, we see here in blue is our flight twin, so Scott, and in green is our ground twin, Mark. And again, we see all the green samples are separate from all the blue samples. We already know that they're significantly different between even at baseline and throughout the entire study. But within Scott, the 4 time points that we know that he's in flight harbor a more similar microbial community than the other 5 time points. We have some statistical analyses. So within Scott, there's a significant difference between the 4 flight conditions, 4 flight samples and the 5 ground samples. Whereas those same time points, 4 versus 5 in Mark, we don't see a significant effect. Now, that's a very high level. That doesn't mean that there aren't any effects in the overall microbial community, we can start to look for features that are microbial features, either taxa or functional genes or pathways that, for example, are low to start with, that go up in space and then return low, or some that start high, stay high the whole time. So we can look for different patterns, and so there definitely are some organisms that are affected long-term by flight, but overall the feeling was that the microbial communities, more or less in Scott, return roughly to baseline upon return to Earth. We asked in a later analysis by David Danko in Chris Mason's group whether there are any specific pathogens that share this kind of up in space, down on Earth, and he did find one putative pathogen, a Serratia species, that basically in the ground twin is kind of low, in the flight twin, that's Scott, is low to start with, and then during our 4 flight samples goes up in relative abundance, but then upon return to Earth, the level of that pathogen returns, a putative pathogen returns to baseline. So it is at least possible that overall there were not a lot of microorganisms, but there are some indications that there are at least some pathogens that do follow this pattern, increase in relative abundance. We don't know what the health implications of this finding are. Okay, so just a quick summary, and then if I've got time, I've got one more slide after this. But again, we more or less see a return to an approximate baseline condition of the kind of global microbial community upon return to Earth after a year in flight. We do see this one pathogen that seems to go up during flight and returns not quite to baseline, but is lower upon return. And as I mentioned before, an N of 1 study at least demonstrates that it is possible that you can live in space for a year and not have a major loss of microbial diversity in your gastrointestinal tract. And that I think overall, one of the feelings from me was that this probably suggests that the microbiome is not the #1 biological concern that NASA should have. Now, there are other studies ongoing with more astronauts, and there does seem to be some variation between individuals. So it's not cut and dry, N of 1. I know that's time, but let me just show you one more slide for 2 reasons. Oops. One, Nico's talked a little bit about his virome project. So I want to show you that we also analyze both the fungal and viral microbial communities here, and that we see broadly similar trends, that the twins have distinct viral and fungal microbial communities from each other. Particularly in the fungal microbial community, we again see this very strong flight effect. The four flight time points look really different from the 5 ground time points, and we don't see the same effect in the ground twin. The virome is a little bit more variable. We do see some clustering, though not significant, of the 4 flight time points. So there's much more to be mined in just an N of 1 study, but we really need a higher replication, and that's why we do a lot of the mouse studies. So thanks everybody for your attention. Happy to take questions.
Yeah.
So you've looked at the microbial community, whether a particular strain has gone up or down. Have you looked at whether any pathogen or a not pathogenic microorganism has taken up virulence upon spaceflight?
Yeah, it's a very good question. It's a little bit beyond both my expertise and the capacity of the study. As I mentioned, there's one. Again, when you just have molecular data, remember, you can only kind of say, it's a putative pathogen. Sometimes you don't know what it's actually doing in situ, in space, in the gastrointestinal tract of the astronaut. But there is some indication that at least this one organism does. But we haven't looked in this study, and I think it's really beyond the data set to see if there's a broader range of increase in pathogenicity. We didn't see at a pathway level or a gene level a dramatic increase, a differential abundance, relative abundance in ground versus flight.
Yeah. So, does this translate into that microbiome? Sorry, diet has no effect at all on microbiome, because I presume that diet of the even astronauts on the ground and in space flight would be dramatically different. So would this translate into saying that diet has no influence on whatsoever on microbial community structure?
I love this question. It's something that I thought a lot about. Before I saw the data, I was sure that the flight versus ground within the flight twin would be so far apart. I was like, they're eating, dehydrated food, and everything is different. So I want to give just two answers to your question. One is, my feeling after seeing the data is that, yeah, they're eating something different, but it's still food. You know what I mean? It's like, it's still made up of the same components of food. So I agree. I was expecting a much larger effect. I don't think you can say food. We know that changing your diet will have an effect on your gut microbial community. The shift that we see in the gut microbiome is the outcome of a multifactorial experiment that we're running on the astronauts, right? So it's not just a change in diet. It's microgravity, which could change the transit of stool through the gastrointestinal tract. It's a disrupted circadian rhythm. It's different carbon dioxide in the atmosphere, different temperature. So there are a million factors that are kind of going into that. I will say that we are sending up Fred and Martha are sending up another mission in February, a mouse centrifuge study where we can at least isolate microgravity as a confounding factor. So they're going to send mice, have them centrifuge both at 1G, 2/3 G, 1/3 G, and microgravity. And that we can at least say, if we still see the shift in gut microbial community, now we know that it's not the gravity alone, it's the other features, space radiation and so forth. But great question.
But we can discuss
Okay, we have to
more
stop
later.
here. We are running late. We can continue the discussion during lunch.
One question.
Great. Thank you so much.
Professor Green.